So today we'll go with another interesting topic, which is about DNA replication. If we talk about the real meaning of this DNA, which we know it's a double helix structure, which is acidic in nature. We discuss this in the packaging of DNA video as well, and a separate video for complete description of DNA. Deoxyribonucleic acid is a double-stranded acidic replication, which means if we just make copy of themselves, or when we just replicate or provide the same thing. So here topic is about DNA replication. This means we are going to make copies of DNA. Copies of DNA. Now the copies of DNA, which mean it is having different types, which we'll discuss. Why is that important? If we just see this DNA, which is simply a parent, right? We have just taken parent, we need to divide it. But what if this is, we know this is double helix structure, right? But still it is not able to divide to the daughter cell by providing both strand. If it can, it will give to this one, so this means this one is remaining. If not possible, this means it's not possible, right? So what they'll do, and even why we require two double stranded for each, because we know if we just take egg, which is taken egg and sperm. Egg is taken from woman, like from female, and sperm taken from male, right? If they both get fused, then what would be formed? Then zygote would be formed. Sperm is simply, right now they are having 23 chromosome. This egg is also having 23 chromosome. When they both get fused, this zygote which will be formed would be having 46 chromosome. This means just because it's important to get 46 chromosome diploid structure by combining two. So if this DNA would be given to this one, what will this DNA do? I mean, there would be no part. So this means zygote can never be formed for 23 chromosome, right? That's why we just say DNA replication is quite important. And DNA replication occur at the phase interphase. During the interphase, or only the S phase of interphase, basically S phase of interphase, it was given by Watson Crick. I'm just highlighting, I'm not writing. This was given by Watson and Crick, model of DNA. What he told exactly, we're going to discuss that as well. So we have different types. Now what are these types? First one called as conservative. Second is called as semi-conservative. And third one called as what? Third one is called as dispersive. Right, these three different types. Now let's see what exactly we need to discuss and what DNA is formed of. So first one called as conservative. In this conservative, what exactly is the concept? In this one, all the things, if you just take this strand, and here we have this strand. One is taken from parental, like from father, basically parental, right? And we just simply say this is taken from parental conservative. In this one, all the strand, all both strand from parental. If this is the parental, right, sorry, if this is the parental one, this will get divided into two other forms, like this one. So this means in this one both are going to be formed new. So this means these both daughter DNA is going to be new. Conservative, both new. When we talk about semi-conservative is like, if we just give an example, which means semi means half. Of course we know that. So this means there would, of course we need double helix. This means we need two strand. First one which would be old, taken from the parental, and the second one that's called as a new. This means how is that? If we just take example, for suppose this is basically husband, and here we have another strand which is basically called as wife. This means these both are from uh, they both are going to get new bond. This means first one, the husband will of course need someone, like of course they need a female, so this means this will get wife. At the time they will become confirmed. Here they need husband, so this means what, why I'm writing this, right? So this is the concept. This means semi conservative, semi means half, so this means half bond, not half bond, but the half one strand would be from the same, which is simply here husband, and here is the wife, which both are same, but the new which they are getting, here's the wife, here's the husband. This means one strand would be same, one same, one different. Same, which means which is already provided, and this will get, I mean, after in the stage semi conservative. So this means when we talk about the DNA basically is semi conservative. Why? Because it get a stand, for one stand they already have. When we make the structure like this one, wait, this is the concept. So here the nitrogenous bases are present and all. So what we see DNA is a semi conservative. When the during the process of replication, one will remain, the old one will remain same, and the second one will get second as a new, because this one will again get pair with another one like this one. So they got new here, even this will pair with another, this will get new. So like that the concept continues. And last one, which is called as dispersive. Even when we talk about this word, this means a mixture. Now mixture of what? If I suppose, if I just make this and this, right? First one, which is basically old, second one is new. So the new DNA which would be formed would be what? This mean basically, if you just understand, this would be mixture of both. Okay, I'm just making here one part, here second, I mean, you can just identify like this is the second part. You can see here's the mixture, here again the mixture, and again, like this cycle will continue. So this, you see, this part is different, this part is different. So in this one, half part would be same, half would be new, dispersive. They will only dispersing themselves. So when let's read it out so that you get it more nicely. So DNA replication, this is about nitrogen spaces occur, the process called as DNA replication, and making copy of itself, the exact copy of DNA called as DNA replication, right? And this occurred during the S phase of the interphase, during the cell cycle, and have so many things. Let's cover this main concept. We have three types: conservative, semi conservative, and dispersive. Conservative was what? Conservative is simply according to this model, parental double helix remain intact. This means the new molecule which is formed will all new copy. Conservative means all new copy will be formed. Semi means half. This means according to this model, the separate each function, template, total cells, and these all. This means one DNA here, parental, which is old one DNA, which and one new DNA would be formed for this one. So we just say some part of parental DNA will remain conserved. This means they will remain same. So one part is going to remain the same. And the last one, dispersive. In this one, what we discuss in this model, all four strand of DNA after replication have mixture of old and new DNA. So this means as I mentioned, they will have old and new both. Now let's check it out in the picture. First one is conservative, if I'm not wrong. Okay, here's the basically picture. First one is semi conservative, second is conservative, third dispersive. In semi conservative, what we observe, what we are going to do, if this is a simple DNA, what we'll see, we'll just see the copy of the two bands. First one is the ring chain like this, and second, which will get fused, or I mean not fused, but the second DNA would be this one. So semi means half, one would be old, one would be new, right? When comes the second one here, conservative. In conservative, what we see the new one, which would be one new, one, or like conservative, which means both would be new. So if you see this, the second is going to be the another, and here both are separately formed, right? Dispersive, which means if you've taken this one, here you see the mixture of dark color, light, dark, light. This means this region continues here even light dark. So yes, like this we can say for the dispersive, half part is that one and half light half dark, like that. So DNA replication ends here. Thank you for watching. Bye bye.